All right, moving along in chapter 12, lesson 12.2 is all about the structure of DNA. And the goal is to describe the basic process of DNA replication and how it relates to the transmission and conservation of genetic information. Far side is yakety yak yak, yak, yakety yak yak yak. That's what cows think we say. Our objectives are to identify the chemical components of DNA, discuss the experiments leading to the identification of DNA as a molecule that carries genetic code, and also describe the steps leading to the development of the double-helix model DNA. So let's think about it for a little bit. The DNA molecule must somehow specify how to assemble proteins, which are needed to regulate the various functions of each cell. So what kind of structure could serve this purpose without changing from cell to cell? Understanding the structure of DNA has been the key to understanding how genes work. So our first objective is to identify the chemical components of DNA. So nucleic acids are long, slightly acidic molecules originally identified in cell nuclei. Nucleic acids are made up of nucleotides linked together to form long chains. The nucleotides that make up the DNA are shown here. DNA nucleotides are made up of three basic components. You have a 5-carbon sugar called deoxyribose, a phosphate group, and a nitrogenous base. All right. The nucleotides in a strand of DNA are joined by covalent bonds formed between their sugar and their phosphate groups. DNA has 4 kinds of nitrogenous bases: adenine, guanine, cytosine, and thymine. The nitrogenous bases stick out sideways from the nucleotide chains. Okay? The nucleotides can be joined together in any order, meaning that any sequence of bases is possible. Here are the actual chemical differences between them if you're going to build these molecules. All right, take a look at this at your own time here. So, objective 1, quite simply, was identify the chemical components of DNA. DNA is a nucleic acid made up of nucleotides joined to get into long strands or chains by covalent bonds. And you need to know all 4 types. So objective 2, discuss the experiments leading to the identification of DNA as the molecule that carries genetic code. So this is the history of how it was discovered. First, we have Erwin Chargaff. He discovered that the percentage of adenine and thymine bases are almost equal in any sample of DNA. The same is true for the 2 other nucleotides, guanine and cytosine. The observation that A equals T and G equals C became known as one of Chargaff's rules. Okay? The next science that scientists need to speak about is in the 1950s. It was a British scientist named Rosalind Franklin. And she used a technique called X-ray diffraction to get information about the structure of DNA, of the DNA molecule. X-ray diffraction revealed an X-shaped pattern, all right, showing that the strands in DNA are twisted around each other like the coils of a spring, as you can see here. The angle of the X-shaped pattern suggested that there are 2 strands in the structure. Other clues suggest that the nitrogenous bases are near the center of the DNA molecule. All right, so now we get to the 2 gentlemen that you've probably heard before. At the same time, these guys, James Watson, an American biologist, and Francis Crick, a British physicist, were also trying to understand the structure of DNA. So they built a 3-dimensional model of the molecule. Early in 1953, Watson was shown a copy of Franklin's X-ray pattern, the woman, British Rosalind Franklin. The clues in Franklin's X-ray pattern enabled Watson and Crick, there they are, to build a model that explained the specific structure and properties of DNA. James Watson and Francis Crick introduced the elegant double-helical model for the structure of deoxyribonucleic acid, or DNA. Let me go back, sorry. So DNA is a substance of inheritance and is the most celebrated molecule of our time. Hereditary information is coded in DNA and reproduced in all the cells of the body. This DNA program directs the development of biochemical, anatomical, physiological, and to some extent behavioral traits too. So objective 2 is to discuss the experiments leading to the identification of DNA as a molecule that carries genetic code. So what happened was the clues in Franklin's X-ray patterns enabled Watson and Crick to build a model that explained the specific nature and properties of DNA. So you need to know all those scientists. Objective 3 is describe the steps leading to the development of the double-helix model of DNA. So the double helix looks like a twisted ladder. In the double-helix model of DNA, the two strands twist around each other like spiral staircases. The double helix accounted for Franklin's X-ray pattern and explains Chargaff's rules of base pairing and how the two strands of DNA are held together. In the double helix model, the two strands of DNA are anti-parallel, which means they run opposites of each other, in opposite directions. This arrangement enables the nitrogenous bases on both strands to come in contact at the center of the molecule. It also allows each strand of the double helix to carry a sequence of nucleotides, arranged almost like letters in a four-letter alphabet. Watson and Crick discovered that the hydrogen bonds could form between certain nitrogenous bases, providing just enough force to hold the two DNA strands together. Hydrogen bonds are relatively weak chemical forces that allow the two strands of the helix to separate. The ability of the two strands to separate is critical to the DNA's functions. So you got to think back to what we know about hydrogen bonds back in the beginning of the school year now, guys. Watson and Crick's model showed that the hydrogen bonds could create a nearly perfect fit between the nitrogenous bases along the center of the molecule. These bonds would form only between certain base pairs, adenine and thymine and guanine and cytosine. This nearly perfect fit between AT and GC nucleotides is known as base pairing and is illustrated in the figure. Watson and Crick realized that base pairing explained Chargaff's rule, and it gave a reason why A equals T and G equals C. For every adenine in a double-stranded DNA molecule, there had to be exactly one thymine, and for every cytosine, there was one guanine. Makes sense. So objective 3 was describe the steps leading to the development of the double-helix model of DNA. The double-helix model explains Chargaff's rule of base pairing and how the two strands of DNA are held together. So our objectives, again, were identify the chemical components of DNA, discuss the experiments leading to identification of DNA as a molecule that carries genetic code, and describe the steps leading to the development of this double-helix model. So make sure you can do that. The history behind DNA, the chemical components of DNA, ask yourself, do you understand all this stuff? If you don't, go back and look at it again, because it all supports our goal to describe the basic process of DNA replication and how it relates to the transmission and conservation of genetic information. I didn't put a cute picture there again. Sorry.